This video is Unit 4, Part 2, or Unit 4B, and this will continue our discussion on evolution. So I want to talk quickly about different evidences for evolution by natural selection. I'm just simply going to list these and give a quick overview of them. The first is the fossil record. And the fossil record or fossils are just preserved remains of past organisms, and what the fossil record does is it allows comparison between organisms of yesteryear and those that are similar to them today, and it can show us links between ancestral species and modern species today. Second is geographical distribution. And this is the idea that organisms from similar environments develop similar adaptations, and we call this convergent evolution. All aquatic animals have fins, even though there are reptiles and fish and mammals in the water. That is an example of convergent evolution. All monkeys in the rainforest, whether they were from Indonesia or from Brazil, have these long tails and have hands that grasp and those sort of things. All right, and so these are all similar adaptations that have developed in similar parts of the world. Another one, very important, is this idea of homologous structures. Now, homologous structures are basically similar structures that don't necessarily have similar functions. Now, oftentimes, homologous structures, this is the picture that is used. Why is that? Well, you can see that these are structures that all are similar. The color coding here kind of shows you that. They all have similar bones, forearm bones. They all have similar, or upper arm bones. They all have similar forearm bone structures. They all have these wrist bones. And they all have these digits. Birds are a little different. But each one of these limbs has a different function, whereas humans have a very versatile function. Horses run, cats do cat things, bats fly, birds also fly, whales swim with theirs. So very different sorts of functions. And so organisms that have similar structures to one another show a common ancestry to one another. These structures have developed different functions depending on what they're needed to have, based on what their environment needs. But you can see here that they definitely they're very similar, but they have a different function based on what their environment needs. Now, this is different than something called an analogous structure, which is to say, like, well, this is supposed to be a dragonfly, really bad dragonfly, really bad dragonfly. Maybe better, a little bit better dragonfly. And that's a dragonfly, and this is a, draw my standard bird that stands here. There's a bird that has a wing, and then, of course, our friends, the bat. It also has wings, but there are three different kinds of animals. The structure has the same function, but it's a very different structure, whereas bird and bat wings are pretty similar. Insect wings are completely different. And so analogous structures typically have something to do with what we mentioned earlier, convergent evolution. Fish and our fins and flippers are very similar to this as well. Another evidence, and this could fall under homologous structures, but we'll give it its own title, is vestigial organs or vestigial structures. And a vestigial structure is structures that are no longer being used, but at some point in the animal's evolutionary history probably was used. Pelvic bones and whales are a perfect example of this. Whales, so here's a whale. Whales have a bone back here in their pelvic region that seemingly attaches to nothing. They have this whole skull thing going on here, and they have a backbone, and of course they have the flippers. But this pelvic bone attaches to nothing, and so except for some muscles. And so the idea is perhaps that pelvic bone and those vestigial bones were at one time the remnants of a leg in recent evolutionary history. Embryology is another one. The way this works is embryos of all vertebrates have very similar structures at their early stages of development. Now, vertebrates, all of this goes back from fish to, say, humans. Well, at their earliest stages of development, at the embryonic stage, the early embryonic stage, you have a very similar looking kind of structure here, where you have a tail, these slits called gill slits here, this dorsal nerve cord, and then with fish, it basically kind of grows and straightens out, whereas with humans, we lose the tail, we lose the gill slits, and those kinds of things. But you can see that we have a similar structural history as far as our embryos are concerned, so that points to a common ancestry. And then lastly, is something relatively new, and this is molecular evidence. This is like DNA, proteins, amino acids, hormones. Organisms that have a very similar biochemistry are going to be more closely related. Now, we talked about natural selection, but I want to talk about the difference between microevolution and macroevolution. Microevolution is small changes in a population uh, over time, and these small changes lead to better adaptations, more fitness. Macroevolution represents large changes that often lead to the creation of a new species. Now, many times, microevolution and macroevolution are separated from one another. Someone will say, well, I see the case for micro, but macro I don't really buy into. Well, macroevolution does not happen without microevolution. And so the two are necessary for one another. And so the common example of macroevolution in animals is fish represent the first vertebrates, and then from fish, the first amphibians have common ancestry, and then reptiles, and then mammals. And so this would show a change, macro evolution change over time, but what sorts of micro changes have to happen in between each one of these? And so that's how we need to think about the difference between micro and macro evolution. Now, when a new species forms, this is called speciation. And a species, first of all, what is a species? A species is a group of similar organisms that reproduce with one another to make fertile offspring. A lion and a tiger can hook up and create the mythical liger, but that liger is unable to make its own offspring. And so a lion and a tiger are separate species, whereas a horse hooks up with another horse and makes other horses that are able to make other horses. Well, that is the difference. And so speciation occurs largely because of a change over time, like we talked about. But what is that change? It is the change in the gene pool over time, not gene poop. That's pretty funny. All right, the change in the gene pool over time. What is a gene pool? This is just all the genes in a given population. And let me just use an example here. So let's say that in a particular population, well, let's go back to our deer reference from the previous video. Let's say that in a population, 40% of the deer have thin coats, and 60% of the deer have thick coats. Over the next 10,000 years, the temperature drops 50 degrees on the average temperature. Something crazy happens on Earth. What would we expect to happen to this population? Well, we would expect to see a change in this frequency because these thin coats are no longer going to be able to survive. And so, let's say 10,000 years from now, we come back and we are 10% thin and 90% thick. This is a change in allele frequency. Frequency is just percentage. Change in allele frequency. What is an allele? An allele is just an expression of a gene. And this represents evolution. Now, there are two men who formulated a principle called Hardy and Weinberg. And their idea was basically that in order for a population to stay the same, to never change, or to be at genetic equilibrium, they had to meet 5 categories or 5 qualifications. And they are these. Population must be large. Little changes in a large population won't show up as readily as a small population. Mating has to be random, meaning that there isn't something that makes someone or an organism want to mate with one other person or one other organism more than another one. And so there isn't the strongest bear in the woods or whatever. It's just everyone is on equal footing. No gene flow. That means that there's no flow of genes into or out of a population. No mutations. So no random changes in the genetic code. And then lastly, no natural selection, which denotes a few things. No limited resources, therefore no struggle for existence, no differential reproductive success. Everyone has the same ability to survive that each other person has. And if that happens, then there is no change in allele frequency. However, situations like this happen. And so Hardy-Weinberg's genetic equilibrium was largely something that doesn't exist in nature. There are instances where this has been observed, but largely it doesn't happen. And so this answers the question, can evolution be measured? Evolution can be tracked by just simply looking at the change in allele frequencies over time. And there are equations to do this, and you can look at different graphs to do this. Now, we simplified this, and we talked about how most populations, whereas this would be a phenotype. And what is a phenotype? It's just a physical characteristic. And this is the number of individuals All right, where most populations, whereas this is one extreme, this is another extreme, you're going to have some individuals that are in that extreme. Most individuals are going to be here in the middle, and then you're going to have some that are in this other extreme. So what do we mean by this? Well, let's just take, for instance, chicken leg length. All right, some chickens are going to have really short legs. Some chickens are going to have really long legs, but most chickens are going to have medium length legs. All right. Now, what can happen in a population like this? Well, let's take our chicken population, for instance, and let's say that in our particular chicken population, something occurs that causes short-legged chickens to be selected against, meaning that something is killing off all the short-legged chickens. Perhaps the water has flooded, or the area has flooded and flooded for thousands of years, and chickens have chosen to live in that area, and it's just tall enough to where it was drowning all the short-legged chickens. I don't know. Something's causing the short-legged chickens to be selected against. And so what would you expect this graph to do? It's going to shift in a direction. And this represents natural selection. And we call this type of selection directional selection. Now, that isn't the only situation that can happen. Let's just say in this particular, we'll use chicken leg length again, because it's fun. Well, let's just say that in a particular population, maybe the medium length chickens are the best. They're the ones that are selected for. Short-legged chickens and long-legged chickens are being left out because they are no longer loved by society and they're kicked out to the edges or whatever. I don't know. Something is causing medium length chickens to be selected for and short and long, both extremes, to be selected against. And so what we call this graph, and I would even want this to be up even higher than that, where there's a definite middle. We call this stabilizing selection. All right. Stabilizing selection. So the way I remember this is that if you stand in the middle, it's more stable. So it's a stabilizing selection in that the middle is being selected for and the extremes are being selected against. Well, let's say that long-legged chickens and short-legged chickens are a novelty, and people like novelties. And so, or people don't like novelties, and instead they pick up only chickens that have the medium length legs, and they go home and have a nice chicken dinner. Well, what would we expect to happen in a population like that? Short-legged chickens are going to survive. So are long-legged chickens. But medium-legged chickens are not going to make it because they're all being taken home and eaten. And so the middle has been disrupted, and this is called disruptive selection.